Dear friends and my dear students, myself Dr. P.K. Verma. Today I shall begin the sessions on human physiology, and in the first session I will discuss anatomy of digestive system. Our food mainly constitutes the plant products, but sometimes animal products like fishes, meat, eggs, milk and curd also are taken. The food constitutes carbohydrates, protein, and lipids, along with a small amount of vitamins and minerals as part of food. The complex organic molecules, such as carbohydrates, proteins, and lipids, cannot be directly absorbed by mouth and stomach. Therefore, we need a digestive system. We have got a very efficient digestive system comprising mouth, alimentary canal, and digestive glands like the salivary glands, gastric glands, intestinal glands, liver, and pancreas. In digestive system, the enzymes are secreted to digest the food. Alimentary canal absorbs them and the blood circulates food from various tissues to be assimilated and utilized. So the animal-like feeding and nutrition includes feeding, digestion, absorption and assimilation. Now come to the first organ. It is the oral cavity or buccal cavity. It is an organ of mastication, taking taste of the food as well as partial digestion of some substances like the starch. We have cheeks and a small mouth lined by lips, so the mouth can be closed, and by movement of the jaw, the food is properly chewed, masticated, mixed with saliva, and then engulfed. The oral cavity is separated from the nasal chamber by presence of hard palate anteriorly and soft palate posteriorly. From the hind side of soft palate, a muscular valve hangs down, which is called uvula. It can spread over the opening of internal layers or the nasopharynx and close them. Anteriorly, both the jaws have a set of teeth comprising four types: incisors, canine, premolar, and molar, and hence the mammals are heterodont. The upper set of teeth is called maxillary teeth, and the lower set is called mandibular teeth. Now, incisors are two in number in each side of each jaw, that is, two pairs of incisors in each jaw. These are chisel shaped and used to cut the food like grasses into fine pieces. These are more developed in herbivorous animals. Canines are one pair of canines in each jaw. These are slightly pointed and used to tear the flesh. These are more developed in carnivorous animals. The premolars. There are two pairs of premolars with slightly flat surface and a few ridges. These are used to grind the hard objects. These are absent in children, and at their place, two pairs of molars are formed. The molars are three pairs in each jaw, but the third pair, called wisdom teeth, grows lately, hence regarded as vestigial teeth. Thus, humans have a total 32 teeth in the adults. You can see the photo. It is the semi-diagrammatic representation of the buccal cavity. The cheeks and skin from right side has been removed. There are three pairs of salivary glands. The first one is parotid gland and its ducts, extension ducts, opens in the upper jaw. The second gland is sublingual gland. It opens under the surface of lingua. The third one is the submandibular gland below the mandibles, and its duct is called Wharton's duct. In addition, there are teeth and tongue in the buccal cavity. Now come to the dental formula. Dental formula is made by using arrangement of teeth and their number in the left side of upper and lower jaw, and the number of teeth in both jaws is doubled to get the total number of teeth in the entire oral cavity. I, C, PM and M. It means incisors, canine, premolar and molar in both the jaw. In children, the number is 2102, 2102, that is 20. They have no premolar. In place of them, there are two pairs of molars. In adults, the teeth are 2123 by 2123, total 32. 20 teeth of children are milk teeth or primary teeth that grow twice in life, so these are diphyodont. And the 12 teeth, two premolars and a wisdom tooth in each side of each jaw, are adult teeth that grow only once, so these are monophyodont. This is the arrangement of teeth in the children, that is primary teeth. Now see the arrangement in adults. These are permanent teeth. You can just compare them in another diagram. Both jaws have been shown in this diagram, upper jaw and lower jaw, in the babies as well as in the adults. Now, there are some examples of other terrestrial mammals. Pig and horse have 3143 by 3143, total 44 teeth. It is highest number in the terrestrial animals, but in the aquatic mammals such as whales and dolphins, the number of teeth exceeds 60. Mice have 1003 by 1003, total 16. They have neither the canine nor premolar. In place of them, a long gap is left that is known as diastema. The rabbit has 2033 by 1023, total 28. In rabbit, the upper jaw has two pairs of incisors. The second pair is behind the first pair. In the upper jaw, there are three premolar, but in lower jaw, one canine, one incisor, and two premolars are present. Cow has 0033 by 3133, total 32 teeth. In the upper jaw, they have no incisor, no canine, but in lower jaw, three pairs incisors, one pair canine, as well as three pairs premolar and molar are present. Sheep has similar formula of the teeth. Cat has 3131 by 3121, total 30 teeth. Dog has 3142 by 3143, total 42 teeth. Teeth are ectodermal, in which the jaw epithelium becomes invaginated and then evaginated to form two layers of cells just like an inverted cup. The ameloblast develop in the growth period on the top to secrete enamel, the hardest material in our body, but in adults, enamel is not formed freshly because there is no ameloblast on the top layer. The inner layer forms odontoblast that secretes bony hard dentin, from which the major part of tooth is formed. Dentin is similar to bone but not the bone. Now see the process of development of teeth. You can see just invagination in the first diagram. Then the lower part becomes evaginated, forming two layers of inverted cup-like structure. Later on, enamel is secreted on the top by the ameloblast cell and dentin by the odontoblast cell. You can see this in diagram D. After development, a small pulp cavity is formed under the coverage of odontoblast. In the pulp cavity, arteries, veins, and nerve fibers are extended. See the earlier stage in which the bud of tooth is developing. Enamel is on the upper layer. It is secreted by underlying ameloblast. Dentine follows ameloblast and it is secreted by odontoblast. Now there are some important points to be remembered. In ruminants, all teeth are molarized to grind hard food such as dry grasses and leaves. In elephant, first pair of upper incisors form the tusk. In walrus, upper canines form the tusk. If a tooth is missing, leaving a space, it is called diastema. Now, let us discuss the structure of tongue. On the floor of buccal cavity, there is a muscular tongue that can rotate around within mouth to mix saliva with the food. It can move into different corners and clean the food particles lying in them. The tongue were evolved mainly to catch the prey and hold food into the mouth. You can see them catching the prey in amphibians as well as reptilians like the lizards. The movement of tongue is under control of glossopharyngeal nerves. On the surface of tongue, there are three types of papillae. The first one are thin hair-like filiform papillae over the upper surface of tongue. They have no taste bud. The second one are fungiform papillae with a small stalk and rounded tips. These are present on the tip of tongue as well as on the sides. The fungiform papillae have taste buds for sweet on tip, salt in the sides, and sour at the posterior margin. On the rear surface of tongue, there are circumvallate papillae which have taste buds for bitter taste. Saliva subserves the sense of taste by acting as solvent. The taste buds can be stimulated only when some sapid, that is tasty substance, is actually in solution. Now see the diagrams related to tongue. In diagram A, distribution of papillae has been shown. The position of taste sweet is at the anterior part, salty in the lateral part, sour in the rear of lateral part, and bitter on the rear surface of the tongue where circumvallate papilla are present. In the middle diagram B, a vertical section of circumvallate papilla has been shown. In diagram C and D, C is diagrammatic, but D is a photograph to show the taste buds having taste pores, sensory cells associated with the nerve fibers. Near pharynx, there are pairs of lymphatic organs behind tongue as well as in the wall of pharynx. These are known as lingual tonsils and palatine tonsils. Their infection is called tonsillitis. In this diagram, you can see the position of pharyngeal tonsil on the upper surface, palatine tonsil in the sides, and the lingual tonsil is not shown, though it is present in the rear side of the tongue. This is the arrangement to show nasal chamber opening behind the mouth where the oral and nasal both parts join together, forming a pharynx. The respiratory tract is present on the ventral of neck. Behind it there is esophagus, that is the first part of alimentary canal, and from back side these are protected by vertebra. Now come to see alimentary canal. The passage of food from pharynx to anus constitute the alimentary canal. It comprises foregut, midgut, and hindgut. The entire canal has inner layer made of endodermal epithelium, but the outer layers are mesodermal in origin. In the foregut, esophagus and stomach; in the midgut, duodenum, jejunum, and ileum; and in the hindgut, cecum, appendix, colon, and rectum are present. This is the diagram showing alimentary canal. Esophagus passes through neck and thorax, joins stomach in the upper part of abdomen just below liver. Thereafter, a small intestine begins as duodenum. You can see jejunum and ileum forming compact coils and surrounded by a long colon, ultimately leading to rectum. There is a small appendix in the right side of lower abdomen. Now let us study the fine structure, that is histology of alimentary canal. The whole alimentary canal is made essentially of 4 types of tissues. These are mucosa, submucosa, muscularis mucosa, and serosa. The first one, mucous epithelium or mucosa. The innermost layer of alimentary canal is lined with a single cell layer of epithelium derived from endoderm. Most of the cells of this layer are columnar, and at places they are larger. They form goblet cells to secrete mucus, so it is called mucosa. In the whole length of alimentary canal, the mucosa layer forms longitudinal folds forming villi. In the stomach region, some shorter folds are formed in between larger folds, so that a large number of villi are formed. In the stomach, the height of villi is shorter as compared to thickness of the wall. In duodenum and ileum, the villi are longer and wall is thinner. These villi are formed to increase the surface area for greater absorption of food. In the large intestine, including colon and rectum, the villi are represented just by the wavy outline of mucosa layer. Second tissue is submucosa, mostly made of areolar connective tissue. It is richly innervated, and the nerve terminals act as primary receptors. These are also known as Meissner's corpuscles. These terminals remain just under surface of the mucosa layer. The blood vessels, nerves, as well as lymphatic ducts pass through submucosa. These nerves are autonomous and comprise sympathetic and parasympathetic both. In mammals, submucosa becomes divided into 2 layers due to presence of a thin layer of muscular tissue in between them. It is called lamina propria. The third layer is muscularis mucosa. In fact, it has 2 layers. There is a thick layer of circular muscles inwards and longitudinal muscles outwards. These are made of smooth and involuntary muscle fibers and act oppositely to each other, that is antagonistic muscles, in addition to strengthening the wall They help in peristalsis and movement of food downwards or backward. In the mammalian stomach and intestine, the muscular layers split and form lamina propria that separates the submucosa into two parts. The parasympathetic nervous system is able to stimulate the enteric nerves in order to increase enteric functions. Conversely, stimulation of the enteric nerves by the sympathetic nervous system will inhibit enteric function and capabilities, mainly peristalsis and movement of food backward. The outermost layer is called serosa. It is made of thin, very thin squamous epithelial cells. It secretes watery materials into the body cavity, hence called serosa. It is made of coelomic epithelium except in the upper esophagus that is present inside the neck. In neck, the esophagus is lined externally by a non-coelomic epithelium known as tunica adventitia. Now, let us study every organ one by one. The first part is esophagus. It is a long tube passing through neck and connects the pharynx with the stomach. At both the ends, it has a sphincter of circular muscles. It has a number of longitudinal pores or villi, but has no specific gland except the mucus gland. The food passes down through esophagus into a stomach. Mammalian stomach is a large reservoir with at least three parts. The upper part towards heart is cardiac stomach, it is a small part. Middle is fundic stomach, it is very big part. And the lower part is pyloric stomach. The fundic stomach is largest part and bend upward, so some part of it come anterior to cardiac stomach. When food comes into a stomach, it fills the lower part first, that is pyloric stomach, and then upper part. Usually in the upper part of fundic stomach, air is filled that comes with food while eating. It is removed occasionally by a process known as belching. This is the structure of the stomach. Its muscles are shown in the upper part, and in the lower part the longitudinal folds forming the villi have been shown. At the end of a stomach, there is a constricted area known as pyloric sphincter. The fine aperture is called pylorus. After that, duodenum begins. In the pyloric stomach, in between the villi, there are gastric glands embedded into submucosa and release their secretions, so these spaces are called gastric pits. The gastric glands have two types of secretory cells in addition to mucus secreting goblet cells. These are parietal cells and peptic cells. The parietal cells, also known as oxyntic cells, secrete hydrochloric acid, and peptic cells, also known as chief cells or zymogen cells, secrete propepsin or pepsinogen. The posterior part of stomach constricts and its opening into duodenum is known as pylorus. It is provided with a ring of circular muscles forming pyloric sphincter. It is not closed except when peristaltic waves pass over it. However, it is a narrow aperture and becomes loose only when food is finely grinded mixed with acid and enzyme in the stomach. See a transverse section of a part of intestine you can see the villi, the folds, finger-like folds on the surface. In the pyloric part, this villi becomes deeper and in between them pyloric glands are formed. Most of the digestive glands are present in the pyloric region. It is a diagrammatic representation showing the gastric pit, and it is a photograph highly enlarged to show arrangement of cells in the gastric glands. You can mark oxyntic cells in the upper part of neck and the zymogen cells near the base edge. In mammals, a definite gastric gland is formed in contrast to many other chordates, where only the folds are there, not a definite gland. You can see the blood circulation, lymphatic vessels, and also nerves passing through submucosa into the villi. Now come to a small intestine. The small intestine or midgut can be divided into three parts in human beings. These are duodenum, jejunum and ileum. Duodenum forms an U-turn and then joins a short straight part known as jejunum. When it reaches within the folds of large intestine, it becomes highly coiled and the ileum is formed. The length of duodenum is 2.4 meters and the length of ileum is 3.6 meters, whereas duodenum is hardly half to 1 meter long. At the loop of duodenum, bile duct and pancreatic duct join the intestine through which bile and pancreatic juice is poured into duodenum. In the duodenum, there are Brunner's glands in between the villi which secrete only mucus, no digestive enzyme. The small intestine has thinner but highly vascular wall. The villi are more numerous and longer. The intestinal glands are straight tubular glands. In ileum, these are known as crypts of Lieberkühn that secrete some digestive enzymes. Mixture of the intestinal enzymes is called intestinal juice or succus entericus. The number of villi and their length increase backward, so these are more numerous in posterior part of ileum, where absorption of digested food material is maximum. In ileum, absorption of food takes place more also due to formation of brush border on the surface of mucosal cells. The brush borders are made of microvilli. In the wall of a small intestine, there are many bunch of lymph nodes known as Peyer's patches. These are used for absorption of glycerol and fatty acids formed after digestion of fat and oil. The wall is supplied blood richly by branches of anterior mesenteric arteries. The blood takes amino acids and monosaccharides from intestine and supplies them to liver through the hepatic portal vessels. The large intestine or hindgut at the site where ileum terminates and large intestine begins, there is a small round cup-like structure known as sacculus rotundus. From here, the large intestine divides into two parts. One part consists of a basal cecum and terminal vermiform appendix form a blind part. In cecum and appendix, there is no digestion or absorption of food, but it harbors bacterial populations that are commensal in most animals, but may be parasitic or symbiotic for some animals. These are vestigial organs in human being. These bacteria help in decomposition of cellulose in these animals. The other parts of large intestine comprises a long colon and a smaller but more spacious rectum. The colon has an ascending, a transverse and a descending limb. A large number of beaded segments